Should we rethink our legal definition of a human embryo? Scientists can now create realistic human embryo models in a lab, leading some to suggest that we rethink how we legally define an embryo. Scientists can now create the embryo-like structures from animal and human stem cells in the lab, and recently researchers unveiled the most advanced human embryo models yet, which resemble natural embryos up to 14 days after fertilization. But this research has raised legal and ethical questions about what an embryo is and how lab-made models should be used. So some scientists argue that the legal definition of an embryo should be changed. In the US, a 1996 amendment that bars federal funding for human embryo experiments defines a human embryo as an organism derived by fertilization, cloning, parthenogenesis, when an unfertilized egg develops into an embryo, or by any other means from one or more human sex cells or diploid cells, which contain a full set of chromosomes. And that definition excludes current stem cell-derived embryo models. But in a paper published August 17 in the journal Cell, researchers argue embryos should be defined based on whether they can become a living organism, rather than how they were made. They propose that a human embryo is defined as a group of human cells supported by elements fulfilling extra-embryonic and uterine functions that combined have the potential to form a fetus. Embryo models that could develop into fetuses would fall into this category and be afforded similar protection as true embryos. The paper authors also want to pin down potential moments or tipping points when an embryo model would legally become a fully fledged embryo. Figuring out this legal definition is important because the technology for carrying these self-organized structures further into development, up to the stage where an embryo becomes a fetus and beyond, could be here within a decade, the scientists say, and without those legal tipping points defined, some scientists would be free to potentially carry these models far into gestation, or even attempt to create a living human from these cells. Now, what are embryo models and why are they being made? Embryo models are typically made from genetically tweaked or chemically treated stem cells, which are either embryonic or derived from adult human cells. Under carefully controlled conditions, scientists can persuade these stem cells to self-organize into blobs of tissue that resemble different stages of early embryonic development, said Roger Sternmy, professor in reproductive medicine at Hull York Medical School, UK, who was not involved in the Cell paper. Models called blastoids can recreate one of the earliest stages of embryo development, a hollow ball of cells called the blastocyte, at around 5 days after fertilization. More complex models called gastruloids model gastrulation when the embryo goes from being made of just one cell layer to becoming a 3D structure around 2 weeks after fertilization. Theoretically, gastruloids could be grown in a lab for more than 14 days, but they generally are not due to 14-day rule upheld by law or by research regulations in various countries. Some researchers, including an influential scientific panel, have called for the rule to be loosened to allow for embryos and embryo models to be grown for longer. These models could provide a detailed look for the 30 days of embryonic development, enabling scientists to improve the efficiency of fertility treatments such as in vitro fertilization (IVF) and uncover causes of early miscarriages. Nicholas Revon, lead author of the Cell paper and principal investigator, said. They could also reveal how development in the womb influences health in adulthood, for example, how it might affect the risk of developing cardiovascular diseases or diabetes, he said. Such results would be technically challenging and unethical to do in a pregnant person, making embryo models especially appealing to researchers studying early development. Why change the definition now? Early definitions of embryos always include eggs and sperm. These ingredients would give rise to a group of cells that self-organize and could form an offspring. But as embryo-like structures become more similar to real embryos, Scientists are realizing that within possibly a decade, such models could, in theory, contain all the elements to give rise to living organisms with no egg or sperm required, Rivdron said. The question that comes now is whether we should adjust the definition based on potential advances in the next decades, as we don't have a clear understanding of when an embryo model would actually become an embryo. Rivdron said he and his colleagues wanted to find tipping points when that line could be considered crossed. One test would assess whether a fetus could feasibly be grown from an embryo model in the lab. Another would see whether similar animal embryo models could develop into live animals when implanted into hosts, and these tests would of course be ethically regulated by authorities, the author said. How might a change affect medical research? These embryo models are a great scientific and ethical opportunity, Rivdron said, and that's because they're easier to produce and study than embryos embedded in a womb, but we have to make sure that all of this is done within a proper framework that fits with societal values. We think that as long as the embryo is capable of forming a living organism, then it should definitely be considered an embryo regardless of how it was made. In their paper, the authors argue that the development of human embryo models in the lab should happen gradually and only be allowed to progress to later stages of development if initial quality checks are passed. They hope that this will help limit unethical research and provide space to reflect on the potential benefits that these models could bring to society. As we further our understanding, like in any field that challenges what's going on before, we have to reflect on what we've known on the basis of the information that we currently have available, Sterney said. It's time to have that dialogue. And this is on Live Science by Emily Cook. So you notice that they want to have a longer time period going into an embryo state. Of course, nobody talks about the fact that there could be a soul connected to this human embryo. Please leave your comments and thank you for your support. Please support my Patreon account. The daily posts are 5 videos daily and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.